Hello everybody, how are we doing today? So today in this video we're going to be going over how vision works. So vision is a very intricate and it gets very complex, but hopefully I can give you a general idea today on how it works. So what we're going to be going at over first, so an overview, we're going to look at the supporting structures around the eye and describe the structure of the eyeball. So your eye moves around in a lot of different directions. So how does that work? We got a bunch of muscles around there and a bunch of associated glands and protective features as well. And then we're going to look at the process of what's known as phototransduction. So light moves as a wave. So it's a particle, has a certain wavelength. Wavelength is peak to peak, and it's expressed in nanometers. So we can see in the visible light spectrum. So as these particles move at this set energy, they actually can cause proteins in our eye to change our shape. So phototransduction, what that word means is taking a photon of light and transducing it and converting it to electrical energy that we can convert for vision then. So really cool process, but as you can sense that it sounds like it's kind of complex. But first here, let's go over some of the accessory structures of the eyeball. Of course, you have your eyebrows and your eyelashes. The eyebrows are to shade the eyes. Eyelashes have a high sensitivity to them for that any debris or anything like that causes reflexive blinking. Um, and then you have your conjunctiva. Conjunctiva is just a protective layering over the eye. So conjunctivitis and things like that would be an inflammation, infection of that conjunctiva. So there's a bulbar conjunctiva and a palpebral conjunctiva. So yeah, the conjunctiva is just a protective layer. And then the cornea is a transparent part of the eyeball. So light can move through the cornea here, through the pupil, which is the hole, and then here would be the iris, which is a muscle pretty much that comes out and controls pupil dilation and pupil constriction. And then we have all the muscles around the eye. So here it's showing the different muscles around the eye. So you move your eyeballs up, you're moving your eyeballs superiorly. So that would be the superior rectus. Down, inferior rectus. To the middle would be the medial rectus, and to the outside would be the lateral rectus. So there are four of the muscles, just up, down, left, and right. And then there's also one that can move the eyeball down and around. That's called the inferior oblique. And then it's hard to see, there's also a superior oblique up here that wraps around this little ligament called a trochlea, and then it connects to the eye, so it moves it up and around. Um, so you have six muscles around the eye conducting its movement, which is kind of cool. Now, there are other accessory features around the eye as well, but these are just the ones we're focused on today. You also have some glands like the lacrimal gland producing tears that run down and keep the eye moist. Also, there's some like antibacterial properties there as well. You have oil glands, tarsal glands, and things like that that are also helping to keep the eye moist because you don't want it to dry out. If you've ever worn your contacts for too long, you know what happens when your eye's not getting enough nutrients and liquids and stuff. Alrighty, so here now we're diving deeper into the eye. So here's a longitudinal section of the eye. So we have, starting back out to what we know here, we have the cornea. So here's the cornea, that's the transparent part of the eye. That's where light moves through. And then so remember then the hole that goes through the middle is called the pupil. And then we have the iris. Remember, the iris is what gives us all different eye colors. So now going around from that, we'll come back to some of the other structures held in here. We have the connective tissue layering. So the connective tissue layering right here, or the white of the eye, is called the sclera. So the sclera is the white that comes up and eventually becomes the cornea. Up here you see something called the scleral venous sinus. This is actually where the fluid in the eye can drain. So there's two chambers to the eye. We see the anterior cavity and the posterior cavity. The posterior cavity is back here, and this would be the anterior cavity over here. There are two fluids in it. In the posterior cavity, we have what's called the vitreous humor, or the vitreous chamber. In the anterior one, we have so anterior A for aqueous humor. Vitreous humor is always there, always steady. Um, Aqueous humor is always being made. So here, this is the suspensory ligaments. It's made from this and rushes out, comes out over the lens, out the pupil, and then drains out the scleral venous sinuses here. Any blockage of that draining can lead to issues, increase of pressure in the eye, pushing back onto the retina, and that's what glaucoma is. It's a failure of drainage of that aqueous humor. All right, that was getting a little to the side. Let's erase some of these lines. So we have, again, the sclera comes back as a connective tissue all the way back, and then it ends up being the connective tissue surrounding the optic nerve. Next layer down is called the choroid. This is the vascular layer of the eye. You see it in red or pink right here. Well, I'm drawing it in red now. It wraps around the eye and eventually becomes the suspensory ligament. Sometimes these are referred to as the ciliary body or processes right here. So the tugging of these ciliary bodies pull on these suspensory ligaments right here, which are holding this structure in place. Kind of hard to see on this image, but that is the lens. So when you look at something close and then look at something far away, your lens is changing its shape. It's called lens accommodation. So there's a pulling of these ciliary zonules on the lens to allow for that accommodation to occur. So that's actually a parasympathetic event taking place right here. The choroid also wraps around as a structure right here. You don't need to know it, but it's called the ora serrata. All right, then we get back into the vitreous chamber. We have the vitreous humor, and now we have the retina layer. So here's the retina. The retina's in yellow right here. So the retina wraps around the eye, and that is where photoreceptors are. So when you have a wavelength of light coming in, a photon of light, it hits the retina, and then that's how you send the signal. That signal then comes out and goes out and back the optic nerve. The optic nerve then travels back to the thalamus, and from the thalamus going to the occipital lobe. So light travels through, boom, hits that. Now, when we look at that retina, we can get a little bit more detailed on understanding what's happening. Now, this is where phototransduction is happening. It's weird because it's backwards. So when this, if we're looking at this part of the retina right here, it's referring to the innermost layer. So it's referring to like this layer right here. Whereas this part of this image up here is referring to the back of it. So when light comes through, light is going all the way to the back of that retina layer. So here, light travels all the way through and hits the back of the retina there. Now, back here, we have what are called our photoreceptors. So we have rods. So here's rods, all in green here. So a lot more rods in this image. And then these ones are cones. So you have one type of rod. That's what you use in dark light. You have three types of cones. You have blue, green, and red, and they see at those different wavelengths. As you can see here, cones are usually 1-to-1, whereas a rod is 3-to-1, meaning if there are 2 points here, if you're trying to separate 2 points, you see them as 1 point down here. You can't differentiate those two. So rods are not, you get blurry vision. Think about when you're walking around your house and it's completely dark, you can't read a book, you can't make out fine objects. But when it's light out and your cones are activated, you can make out those 2 points from each other. Also, you don't want the light to scatter. So back here, there are little melanin granules. Melanin is a color pigment in this epithelial layer back here that helps prevent light from coming back and scattering and activating something over here. Now, also in the daylight, your rods are inactivated because they are known as what's known as bleach. They are very light sensitive. Your cones are what are activated. And then in dark, the rods are activated. And also, like, so you have light and dark adaptation. Say you are sleeping and someone turns turns on all the lights and wake up. Yes, you're glaring, you're squinting, then everything eventually comes into play as your cones reactivate. But now imagine turning all the lights off. Everything is dark for a lot longer. It takes you a lot longer to reactivate rods than it does cones. But cones are known for color vision, and rods are known for black and white vision. Now, the process of phototransduction, what happens if we activate this cone here? There are all these proteins found in this photoreceptor layer, and when light hits that and activates it, it sends a signal down through first a photoreceptor and then a unique neuron here called a bipolar cell, and then from that bipolar cell it becomes a ganglion cell, and then that ganglion cell then becomes the optic nerve and goes back. Now, if you look at the retina up here, so the process, let's zoom in extra far on this image. So the direction we would be going, we'd be coming out, going like this, and then going back. So that means you have a spot right here where you don't have any rods or cones. That spot is your blind spot. So we all have a little blind spot there where we have no photoreceptors. Also, your eyeball has a spot like right in here called the fovea centralis. So the fovea or the macula lutea is an area that you have pretty much all cones. So all cones would be a very, very high acuity vision spot. So that's where you can differentiate the most dots from each other. So can you differentiate these two dots, and so forth? That's very, very highly fine-tuned right here. Whereas out here on the periphery of the retina, that's where you have more rods. And so you have more cones over here. Also, there's vasculature, there's arteries, veins, and so forth found in there as well. Alrighty. And all this, that blind spot then, this is known as the optic disc right here too. Now, this is what that retinal layer looks like on a histology image. So right here is that melanin pigments. Here are the rods and cones. So you can see the outer layers. Here are the nuclei. Here are these parts. These are the bipolar cells up here communicating, and then coming down here to the ganglion cells. Ganglion cells are sending the optic nerve out. So you can see these layers based on where the nuclei are compared to where the nuclei are in these layers here, because nuclei stain blue typically in histology image. So wherever you see that blue layer here, you have a layer of nuclei. Alrighty, so what's happening in the ends of those photoreceptors? So here you have the rods. We have these stacks of like pancakes or discs here. And these discs are constantly being fragmented off here too because light is destroying them. These discs are embedded with a protein called rhodopsin. This is particularly for rods. There are 3 other opsins that we have for cones then as well. And the way this works, light hits it. So here's a photon of light hitting this. And there's a chemical in here called retinal. And that's what this structure is right there. That's called retinal. You can see that down here, 11 cis retinal. So this is derived from carrots. So when they say carrots are supposed to help your vision, this is where that saying comes from. Well, basically what happens is when light hits this arm right here goes from that and goes straight. That's all it does. It goes from 11 cis to 11 trans. And when it does that, it causes a signal to be sent. And that's phototransduction. So the photon of light can cause that event to happen at that particular wavelength, and that then sends the signal. So kind of wild how that works. So here are the rods. Rods are most active at 498 nanometers. That's just a wavelength. And then you have blue cones, you have green cones, and you have red cones. So why can or how can you see so many shades of light? Well, it deals with the percent of absorbance over here. So absorbance is how well those proteins absorb at that particular wavelength. Because you don't only see blue, green, or red, you see all the different shades in between, browns and light greens, red, like light orange, and you know what I mean. So let's imagine you're looking at a color right here. You're looking at this color in the visible light, 500 nanometers. Well, what happens is you activate blue cones at about 20%. You activate red cones at about 50%. And then you activate green cones right here at about 60%. So your brain takes this 20, 50, and 60 and says, oh, it's light blue or green blue. And you're able to do that across this whole spectrum, and that's how we can see the visible light spectrum. If you go higher than this, that's where you get into different energies like infrared. Snakes have opsons that can see in infrared, and that's the heat signature. So some animals have different cones and so forth. So we are known as what's trichromatic vision, meaning we have three cones. Some are quad chromatic, some are just dichromatic, and so forth. I could get into this whole story about where green and red cones came from and evolution wise, but we're not going to get into that here. That's you can take my genetics course if you want to learn about that. So that's vision. And that's vision in as simple terms as I can make it. Um, it's complex. But the idea is that we take this wavelength of light and we convert it to electrical energy. When it activates these photoreceptors, what happens is that it converts it to an action potential, which can then go back our neurons through our optic nerve to our brain and form an image. And in our brain, we form any associations we might have with that particular image. So, that is the little summary of vision here. And like I said, well, like most of these survey lectures, this is just the tip of the iceberg. So if you have any other questions or any questions, feel free to reach out to me and I'll be happy to discuss them. But if not, hope you have a great rest of your day and bye bye.